Okay, good afternoon. Thank you very much for the opportunity to tell you about our work here, and thank you for the invitation to present here. I'm a professor at MIT, and my lab works on developing molecular technologies, especially with applications for gene therapy and genetic medicine. So I prepared three short stories to tell you about. These are things that we've been working on and are quite excited about, and the theme of all three projects that they derive from our study of biological diversity. So, there are thousands, millions of different types of organisms that live around us, and many of them have evolved very interesting molecular mechanisms that we can then co-opt for therapeutic applications. So let's talk about the first one, and this is a system called diversity generating retroelement. So why are we interested in diversity generating retroelement, and how do we sort of come to study it? So one of the things that we have been very interested in, especially coming after studying TAO systems and CRISPR systems, is what other natural systems are there that are easily reprogrammable. So if you think about Cas9, Cas9 is a system where you can rationally design a new guide RNA to then be able to recognize virtually any DNA sequence. And that ease of customizability, that programmability, is very powerful. And so one question that we'll ask is, what else are there in nature that may be customizable or easily reprogrammable so that we can target a variety of different substrates? So if you think about reprogrammable systems of the natural form, one of the key features is that they have a region that is hypervariable. So if you look at a CRISPR protein, a CRISPR RNA, or if you look at a tail protein, you see a feature where there is constant sequence preservation, but then within a very short defined window, there is enormous amount of sequence diversity. And that diversification is how nature evolves different activities. And in this case, we are looking for that particular signature as an indicator of reprogrammability. So here is just a diagram that illustrates this concept. So if you have three different proteins, and they are programmable to recognize different substrates, then the region that is responsible for recognizing that substrate would be the region that is diversified. And so if you sort of align these three different sequences and look at their conservation, you will see that the middle of this protein increases in sequence diversity, because that's the signature for diversification. So then we carried out a computational discovery project to see what other systems are there in the bacterial genomes that have this level of diversity. And so quite easily, we were able to find CRISPR systems. So if you look at the CRISPR spacers, you can see that the spacer regions, so these are the sequences that specify new DNA targets, is very rapidly diversified. But then what flanks these diversified regions are very well conserved. And so it's this kind of step function feature that we're looking for in the sequence analysis. So what else did we find besides the CRISPR systems? Here is one. This is a diversity generating retroelement. And so you can see that if you line up this sequence over here, you'll find that there is a particular position that is very strongly diversified. And so this suggests that this protein here, although is mostly conserved, has a specific region that probably nature changes from time to time to be able to recognize different substrates. And it turned out that this is a protein, it's a lectin protein, so it usually binds to polysaccharide. And so this diversified region probably is responsible for recognizing different sugar substrates. So here's another example of something that is rapidly diversified. Here is a bacterial locus. There's an integrase. And right next to the integrase, there is a non-coding region, so not protein coding, that is also diversified. So if you look at the sequence alignment, you can see that this middle region here changes very rapidly, whereas the flanking regions, including the proteins, are not diversified. And so this suggests that there may be something that's interesting that's happening here. So anyway, so we carried out this computational study, and we identified a number of different systems. So we picked the top 13 systems that show this type of rapid divergence within flanking conserved sequences. And we put them into E. coli to see which one of these systems diversify its sequence. So it turned out that most of them had very little activity in E. coli, but one of them, system number 10, exhibited very robust diversification of that region. And so that told us that this is probably an active system, and moreover, this diversification mechanism is also active and reproduced within the E. coli system. So what is system number 10? This is system number 10. So this is a diversity-generating retroelement. And so if you look at this is the canonical structure. So there is a gene, a lectin protein, and then this protein has a region that is variable, that's diversified, called a VR. And then following this protein, there is a sequence structure within an uncoding region. It's a hairpin. And there's a gene called AVD, followed by a stretch of sequence that is more or less homologous to this region, but not identical. This is called the template repeat, TR. And then it's followed by a reverse transcriptase enzyme. And so this system has been studied by Jeff Miller more than a decade ago. And so the basic function of this system is that the reverse so this TR domain is transcribed into RNA. And then the reverse transcriptase recognizes the TR transcript and then carries out reverse transcription to form cDNA. And this is an error-prone reverse transcriptase. And it only mutates places where there is an adenine. So if it sees an A, it will substitute one of the other three bases in that position. So it's a random mutator that is targeted to the A positions within the RNA. And so this cDNA, after generation, gets homed back into the lectin protein so that the recombined product results in a lectin protein that has some diversified sequence. And so this is a very clever mechanism that bacteria use to be able to diversify these surface proteins through this error-prone reverse transcription mechanism. So this system was discovered in a bacteria called Bordetella, but all of the systems that have been identified to date were not able to get transferred into E. coli, which made scientists speculate that maybe there are host factors or some other enzyme that Bordetella carries that's important for DGR to function. But what we ended up finding is that this system, the system 10 DGR, was able to get successfully translated into E. coli. And so this is sort of more sequence alignment showing how this can get diversified. And so this is the template repeat RNA. And you can see that many sequences of the cDNA coming from reverse transcription has mutations in places where there's an A. So it gets substituted with the other bases. So this becomes a very useful mechanism. And so this is something that we're now exploring to see, can we use it to engineer a continuously evolving system so that we can diversify protein function and then select for specific features. We did some more studies of this system. It turns out that you do need to have at least a 5' homology region in order to get any diversification. So this further shows that it's the template repeat that gets transcribed, and the template repeat has these flanking regions that needs to match at least one of the two ends of that diversified region in order to start to see these diversification. And so this is something that I think shows that the basic mechanism is one that there is homologous recombination. And then moreover, you can mutate this protein to change the base substitution specificity. So this is where the wild type is. And we found this one particular mutant, K71A. And what you see is the wild type, it substitutes for most of the different bases. But then K71A can reduce the substitution rate for these three bases, but maintains substitution for thymine. And then there's another mutant in the reverse transcriptase, I184S, basically changes it to a single substitution. So rather than substituting an A into all three other bases, it's focused on substituting that base with the cytosine. And so this also shows that it's a reverse transcriptase scaffold that we can then evolve to be able to make specific types of substitutions in a target substrate. So that's just one example of exploring natural diversity. Another thing that we have been looking at are within natural diversity, what other types of immune systems are there in the bacterial sort of world? If you think about it, bacterial immune systems are very powerful. Restriction enzymes, which are important for recombinant DNA, is one form of anti-defense or antiviral defense system. CRISPR is another example. And so what we ask is, if we look into the bacterial world, can we identify any new ones? And so this is a summary result from our search. And what we found is that even though there's quite a few known defense systems, but quite easily we can find many previously unknown defense systems. And if you look at these different defense systems, they're very interesting. So they have reverse transcriptase domains, they have different deaminase domains in other systems. So overall, it represents that biochemical activity, a quite diverse set of biochemical activity, can all get co-opted to become a part of bacterial antiviral defense. And so this also shows how diverse they are, and we're now working on trying to understand some of them, and then maybe harnessing some for interesting sort of modulation in eukaryotic cells or therapeutic applications. So that's the first one of looking at biological diversity in terms of defense systems. Another project that we've been working on recently is looking at other ways to develop delivery systems. So right now there are many different ways to deliver therapeutic molecules, biological molecules. There are lipid nanoparticles, there are viral vectors, and those work, but they usually are successful only for a limited number of tissues. You can target liver, the eye, and maybe blood cells. But the vast majority of other tissues is very challenging to target. And so what we did is we tried to explore, rather than viruses, we studied virus-like particle systems. And so what we studied is something that's based, that is related to retroviruses, but both retroviruses and coronavirus and influenza viruses are RNA viruses. And so these two viruses, they don't integrate into their host genome, but retroviruses do. And so what that means is that over the past 50, 100 million years, our evolutionary ancestors have been infected by retroviruses over and over again. And so if we look into our genome, we can find examples of some of these previous infections. So what do you look for? So it turns out that a retrovirus has a genome of the following structure. It's got some non-coding sequences at the ends, but it's got three main protein coding genes. One called GAG, which forms the structural elements of the virus. So that's the virus protein capsid that encapsulates all of the material inside. And then there's the PO gene, which are the enzymes that's responsible for replication and integration of the viral genome. And that's the enzyme molecules inside. And then the third one is the envelope protein, which are these membrane proteins that decorate the surface of the virus. They're responsible for binding to host cells and then facilitating entry into host cells. So that's those spike proteins on the outside. So if you look into the human genome, and others have, this particular computational paper from more than 10 years ago, did an analysis of the human genome to see what proteins, especially native human proteins, carry strong sequence homology to the viral GAG protein. So that's the structural protein. And so the study found three main families. There is one family called the sushi family, which are genes like PEG10, MAR4, RGAG1, RGAG4, so forth. And these are named sushi because they came from, or they're most closely related by sequence to the sushi family of retrotransposons. And then there are two other families, the SCAN family, which is different, and then also the paraneoplastic family, which is the third family of retrotransposon-related proteins. If you look at these proteins, you can see that they are very similar to the retroviral structural protein, the GAG. So GAG has two domains. There is a N-terminus and also a C-terminus, the two blue circles. And so if you look at the sushi or the SCAN or the paraneoplastic families, they all contain very well conserved segments of this capsid protein. And so a few years ago, there were a couple of studies that showed that one of these proteins, ARC is right here, is able to form virus-like particles in neurons. And so we thought if one of these proteins can form virus-like capsids, maybe we can look at these entire families of proteins. Maybe there are actually quite a few of them, and maybe some of them we can repurpose to package therapeutic payloads. So we studied a number of them, and we expressed them in E. coli, and we purified it. And so these are 5 examples. What you see is that many of them, like MOAP1, RTL1, or PMA3 or PEG10, they form these virus-like structures out of the in vitro purified protein, recombinant protein. But not all of them form virus-like structures. So ASPRV1, for example, doesn't form these virus-like structures. But this is a good starting point because most of them form viruses or virus-like particles. And so we studied these different ones. We put them into human cells, and then we took the supernatant and we asked, are these virus-like particles released by the cell and secreted into the extracellular media? And what we found is that 4 of them released into the media, and one particular one called PEG10 releases quite robustly. So there's a lot of it that you can find in the extracellular media. So if these are virus-like capsids, one of the questions that we had is, do they package anything? If they are getting secreted, are they releasing anything from the cell? And are they transferring anything to other cells? So we took the supernatant and we did RNA sequencing to see what's inside. It turned out that the predominant RNA in the PEG10 capsids are the PEG10 mRNA, which is kind of interesting, but if you really think about it, it's not surprising. Because if PEG10 is a viral, if it's derived from virus-like systems, those viral proteins, so the ancestors of PEG10 naturally carry their own viral genome. And so the PEG10 mRNA is kind of the genome for PEG10. So seeing PEG10 getting packaged is actually kind of nice to see. Confirms that they may be binding to their own self-RNA. We analyzed these RNA more thoroughly. And what we found is that there are specific segments of the RNA that interact with the PEG10 protein. And those regions are located in the 5', the 3' UTR, the 2 UTRs, as well as in the middle segment of the PEG10 gene. So these are binding motifs that interact with the protein. So based on that, we thought maybe we can take advantage of these packaging motifs to engineer other synthetic RNA molecules for loading into the PEG10 capsid. And so we engineered a system where we have 3 different elements. There's a fusogen, which decorates the surface of these PEG10-like particles, and then a PEG10, which makes the protein to form the capsid. But then we engineered the RNA as a cargo RNA by taking a gene of interest and flanking it with the 5' and 3' UTR for the natural PEG10 mRNA. So the idea is that these UTRs would allow this new synthetic RNA to interact with PEG10 and get loaded into a PEG10 virus-like particle. So we produce these virus-like particles and then put them under EM, and what you can see is that they form these very nice, very much sort of retrovirus-looking particles from cells. This is a negative control. These are exosomes secreted by the same cells. You can see they number one have a different size, and also shape-wise they're also quite different. And so this allows us to make specific types of particles. We then put these particles onto cells, and we found that they can functionally deliver the mRNA payload. And we also try to pseudotype these virus-like particles with different fusogens. Researchers commonly use VSVG as a fusogen for pseudotyping lentiviruses. But then, so we found that VSVG can work. But then we replaced VSVG with 2 other native human proteins, the syncytin proteins. And we found that these can also pseudotype the virus-like particle and still achieve functional delivery. And so what this condition showed us is that you can actually make a particle that's entirely endogenous. Human protein as the fusogen, human protein as the capsid, and then it packages a RNA that's produced by the producer cells. And so this is one way that we can go about making these sort of virus-like particle delivery systems. So then as next steps, what we're focusing on is really engineering these systems so that they can package larger and larger payloads and then also be able to target different cell types so that we can go beyond the liver, beyond the eye, and then also get into other organs in the body. So finally, the third story is some of our continued effort to discover systems that have applications in gene editing. And to do that, rather than looking for other CRISPR systems, we went back into time and we looked at ancestral systems that gave rise to CRISPR systems. And so what I'm showing here is a phylogenetic tree. And this giant tree shows not only the CRISPR-Cas9 proteins, but also all of the ancestral proteins that gave rise to Cas9. And what we end up seeing is that this slice of the pie, this blue section, these are the Cas9 proteins. You can see how they kind of evolved over time. But then this Cas9 family protein really is just one emergence of a branch of protein out of this much larger family of proteins called IACb. So in fact, Cas9 can be thought of as a subtype of IACb. So what is IACb? What is this really diverse set of proteins? So it turns out that IACb has been known for a long time. It's one of the most abundant genes that you can find in bacterial genomes. Sometimes within a single bacterial genome, you can find 60 or up to 100 different copies of IACb. So it's really one of the most common, sort of most widely spread proteins in biodiversity. Another thing is that IACb has been thought of as a transposable element because it's flanked by repeat elements that are often found to demarcate the ends of transposons. So IACb is the gene that's encoded in this repeat flanked region. If you look at a protein sequence for IACb, you'll find that it shares quite a bit of similarity to Cas9. So on the right is Cas9. So IACb has different domains. So it has this HNH domain, which is a nuclease that's found in Cas9. It also has these 1, 2, 3 domains that are also found in Cas9 that represent the 3 split fragments of the RUFC nuclease domain. And so essentially, IACb is probably a nuclease. It's got a HNH nuclease and it's got the RUFC nuclease, which are the 2 nucleases found in Cas9, and also arranged in this same sort of organization. But there are some differences. It's much smaller than Cas9. It's about a third of the size of a Cas9 molecule. And then also, it doesn't have the rec domain, so that's not there. But it's got a new domain called PLMP domain. So that's the black sort of P rectangle over here. So Cas9 was known to work with RNA. So it's an RNA-guided endonuclease. It uses a CRISPR RNA to specify the DNA target, and also a tracer RNA that forms a duplex with the CRISPR RNA to then get loaded onto Cas9. But it wasn't known whether or not IACb also uses an RNA, whether it's RNA-guided. And so we did some RNA sequencing experiment. And what we found is that there is a conserved, so highly sequence conserved, but non-coding region upstream of IACb that is also expressed as RNA transcripts. And then what is also really interesting is that this conserved region covers this repeat element. So this is the demarcation of what people thought to be a transposable element. So it contains this portion, but it's also transcribed beyond this conserved portion. So there's also a little bit of extra sequence. So we hypothesized that this is probably the guide sequence here. So this is constantly changing so the IACb can target different DNA sequences. And then the conserved region here is the scaffold. That's what the IACb protein binds to. So we tested that hypothesis and turned out that indeed IACb, just like Cas9, is RNA-guided and it cleaves DNA. So first of all, it has a motif that's downstream of the target site, just like you find a PAM sequence downstream of the Cas9 target site. And then the two nuclease domains are individually responsible for cleaving one or the other strand of DNA. And so the RUBC domain cleaves the non-target strand, and the HNH domain cleaves the target strand. So you can see inactivation of them turns IACb into a nuclease. And so this is really nice to see, because it shows that IACb is an RNA-guided endonuclease, but even more importantly, it's a much more compact RNA-guided endonuclease, which makes it potentially more useful as a therapeutic molecule, because it's smaller, it's going to be easier to deliver, and then we may be able to further engineer the RNA scaffold for enhancing different functions. In addition to IACb, we also found another RNA-guided endonuclease, and that's called TNPB. So TNPB is also RNA-guided. It's got a guide molecule downstream as opposed to upstream. And this is also RNA-guided, and it targets DNA. And if you look at the sequence for TNPB, it's more closely related to the Cas12 family of CRISPR proteins. So these two together, which is what we call omega systems, they are sort of the ancestral elements that gave rise to the two different DNA targeting CRISPR systems. So those are the three stories that I prepared for today, but we're really excited to continue and explore this astronomically or exponentially growing biological diversity. So these are sequences, number of genomes that have been sequenced from the bacterial world, and you can see that it's really growing very rapidly. So I think we probably haven't even scratched the surface of this natural diversity in terms of discovering and harnessing useful biomolecules for therapeutic applications. So I think this is a really exciting direction. So last but not least, I just want to acknowledge my lab and collaborators, funding sources, and if you have any questions, you can feel free to contact me as well. So thank you very much. Happy to answer any questions.
So thank you for discussing 3 of your projects with us.
Okay.
We got a number of questions here. I read them for our online audience. What's the current size and delivery limitations to the VLP drug delivery system, if any? Can this cross the blood-brain barrier?
Yeah, thank you for the question. So in terms of these VLP with PEG10, the packaging capacity is similar to what lentiviruses can package, because what you can package is limited by the size of the capsid. So somewhere between 8 to 10 kilobases is kind of the upper limit for what we can stuff in there. We don't know whether or not it can cross the BBB yet. I think we're just getting started with some of these in vivo experiments. But it's possible PEG10 may or may not. But then there are also almost 83 more proteins in the human genome that form these virus-like structures. And so some of them may have that BBB crossing property.
Okay, next question. What do you think the purpose of these viral-like particles in humans are there or can be? Can it be applied to cell therapy?
I think these are really interesting particles. The specific function is not really known. In fact, the biological function for all of them is not really known at all. One of them, PEG10, is an essential protein. So it means that if you knock it out in mice, you will not get viable birth of young mice, young pups. And the way that people have studied them is by looking at development. So there's a group of Japanese researchers who have been studying PEG10, and they found that PEG10 is involved in the development of the placenta. And so when they knock out PEG10, the placenta doesn't fully develop, and so the fetus, the embryo, dies around embryonic day sort of 11 or 12 for mice. And so what that means is that it's involved in placental sort of formation. It may be functioning. Its function may be related to another protein, which is a syncytin. It's involved in forming these syncytial membranes that line the placenta. So that's all we know about PEG10. The other proteins are even less well understood, but I think it's certainly an important area, especially since some of these proteins are essential.
How are you currently transfecting your payloads into PEC-10? And now the question just moved. And what are some limitations or opportunities to improve the manufacturing of these virus-like particles, I guess? So that's the question from Paul. Yeah.
Right. So right now we have been making these PEC-10 virus-like particles using 293 HEK cells. So we introduce into HEK cells the 3 different production plasmids, and then the particles are self-assembled within those cells, and then they're secreted by the producer cell. I think one of the major challenges is to figure out how to scale the manufacturing of these proteins, and that's something that we're working on right now.
How you see ERNs being able to insert DNA segments greater than 50 bases? Must be kilobases.
Yeah, that's a good question. So I think this is an active area of exploration by many researchers. So people are looking at transposable elements, integrases, recombinases, so things that hop around in the genome, and trying to sort of co-opt them for DNA insertion. I think it's a very promising area to look, because that transposable element diversity is enormous, and some of the transposons can be even much larger than 50 kilobases, can be 100, 200 kilobases long. And so understanding the enzymology for some of those reactions, I think will be very helpful for developing new insertion technologies.
Okay, there are a bunch more. Let's go on. We still have a minute or so. What are your thoughts on systemic delivery of messenger RNA therapeutics that can reach specific cells effectively?
I think it's a very exciting direction that really sort of needs a lot of development in. I think we have all, or probably most of us, have experienced the power of mRNA therapeutics through the COVID vaccines, which gave us protection against coronavirus. But then what is challenging is we need to have more delivery vehicles to get RNA into other parts of the body so that we can target them. But there is exciting progress that's being made, exciting clinical successes in liver, in sort of blood applications, and I think as we develop more delivery modalities, I think we're going to see many more interesting, exciting applications.
So another question on packaging. What is the packaging capacity of PG10 EVLP? How does it compare to AAV or lentivirus?
Right. So the packaging capacity is similar to a lentivirus. So it's somewhere between 8 to 10 kilobases of RNA that you can package in. So that's larger than AAV. AAV is around about 5 kilobases of single-stranded DNA. So that's the difference.
So we have more questions. We have a break coming up, so we have some time.
Okay.
Do you still have time for more questions?
Sure. Yeah, I can answer more questions.
So let's do a few more. How do you see the precision of ERNs being increased?
Yeah, that's a good question. So I guess the question is about how do we get these sort of mobile genetic elements to be able to be more precise in where they target. I think part of it really depends on understanding the natural targeting mechanism. So if they have specific mechanisms for homing to safe harbor sites, you might engineer those mechanisms. Some of them are going to be RNA guided, maybe some of them are protein mediated DNA recognition. So I think that's really the key, is understanding how that specificity is naturally specified and then engineering them. There are certainly opportunities to combine those systems with RNA-guided endonucleases like Cas9 or Cas12 to be able to get retargeting.
Okay, let's do one more and then we'll stop. What is this teaching us about how to engineer or search across biology for new tasks, such as material processing, fabrication, sensing, etc.?
I think really there's a lot that's very worthwhile to explore in nature. Very little has been explored so far, so I think there's a lot that we can do. What that also means is that we need to come up with better processes. So far, the way that people have discovered new systems, a lot of it is based on serendipity. One researcher stumbled upon an interesting system report an interesting feature, and then more people study that. And that process can take maybe a decade for it to go from the initial discovery to turning into a technology. So especially as we are getting better at predicting the function of protein sequences and even predicting the structure of protein sequences, that process can probably get accelerated much faster. And so by first rapidly or systematically predicting the function, and then be able to set up high throughput sort of biochemical assays to verify those functions and then optimize them, I think that will be really exciting for accelerating this whole field. So if there are tens of thousands of different mobile genetic elements, you can imagine a way where they can all be identified computationally and then also at the same time experimentally investigated using small volume, high throughput biochemical assays.
Great. Thank you very much for coming, for speaking to us, and for taking so many questions.
Thank you.